Hey, welcome YouTube. I'm Aniket and you are watching Vet Study. So today I'm going to discuss about the genus Staphylococcus. This Staphylococcus genus is very important. So we will discuss this genus in detail. And after discussion, I will show you some images which are very important. So let's begin with the general characteristic. The general characteristic of Staphylococcus are as follow. That Staphylococcus is a gram-positive cocci. So it is very important that this is a gram-positive bacteria, right? And the approx diameter of Staphylococcus is 1 micrometer, and it resembles the bunches of grape. So the name is the Staphylococcus because the name is derived from the Greek word. The Greek word is Staphyl and Coccus. The Staphylococcus means bunches of grapes or berries. Okay, so the name is as follows, Staphylococcus. Okay, after this we have fourth point that there are only 30 species and they live as commensals on the skin as well as mucous membrane. They also act as opportunistic pathogen and they cause pyogenic infections. They are mostly facultative anaerobe and catalase positive. So this is the general consideration, but we have some exceptions like Staphylococcus aureus, subspecies anaerobicus, and Staphylococcus saccharolyticus. These two species or subspecies, you can say, they are anaerobic and catalase negative. After this we have they are generally coagulase positive, and this coagulase positiveness is associated with the pathogenicity. So all those you can say the species which are coagulase positive are generally pathogenic in nature, and the example of coagulase positive Staphylococcus are Staphylococcus aureus, intermedius, and hiacus. Now move to the habitat. As we have discussed that habitat is comparatively they are generally live as commensals on the mucous membrane of upper respiratory tract and lower urogenital tract, and they are comparatively stable bacteria, and they live on skin as commensal, and their distribution is worldwide in mammal as commensal. After this we have the differentiation of Staphylococcus species, because this differentiation is very important. Why? This differentiation will tell you how you can differentiate between the different species of Staphylococcus, or maybe from different genus like Streptococcus or Micrococcus. So basically, how you can differentiate between the Streptococcus and Staphylococcus? Staphylococcus is generally catalase positive, whereas the Streptococcus are negative, catalase negative, right? Staphylococcus species are usually categorized by their colony appearance, hemolytic pattern, and biochemical profile. Apart from this, the ribosomal RNA gene restriction pattern is also used to differentiate or categorize them. And Coagulase test is also important. So these 5 things are very important to categorize the several Staphylococcus species. So we will talk one by one. The first is the colony characteristic. The colony characteristic of Staphylococcus is as follow, that the colonies are white and opaque, and they are 4 millimeter in diameter. Some Staphylococcus species show golden yellow colonies, and they are from the bovine or human strain of Staphylococcus aureus. Next we have hemolysis. So hemolysis, this point is very important, and your teacher may ask question from this point. Hemolysis. So hemolysis is used to differentiate between the aureus, intermedius, and hiacus. These 3 species are differentiated on the basis of hemolysis, and Staphylococcus genus produce 4 type of toxins. These are alpha, beta, gamma, and delta. These 4 toxins are produced by Staphylococcus genus. So these toxins are different antigenically, biochemically, and their effect on the RBC on different animal species, and on the basis of them we can differentiate different species. Like animal strain of Staphylococcus Staphylo intermedius usually produce both alpha and beta toxin, and so we will discuss what is actually the hemolysis. So basically, on ruminant blood, this is the bacterial colony, and these bacteria produce 2 type of hemolysis. The first is the complete, and second is the incomplete. The complete hemolysis is produced by alpha hemolysin, or you can say the toxin, and the incomplete one is produced by beta hemolysin. This alpha produce narrow zone of hemolysis, and this produce wider zone of hemolysis. This question is very important. you should not, skip this question. This double hemolysis is very important. Remember this thoroughly. So what I'm saying that complete hemolysis is produced by alpha hemolysin, and this produce narrow zone, and beta hemolysin produce wider zone, whereas the hemolysis is incomplete. Simple meaning is that in complete hemolysis all the RBCs are hemolyzed, but there is narrow zone. This zone is narrow comparatively, but where beta hemolysin are present, they will produce the incomplete hemolysis, means not all the RBCs are hemolyzed, and this will produce the wider zone. Okay, and this complete phenomena is known as double hemolysis, and this will produce a characteristic appearance. And after this we have Staphylococcus aureus, which are non-hemolytic. So this is also an important point, that Staphylococcus aureus are non-hemolytic. Okay, and delta and gamma. Delta produce incomplete, whereas this gamma produce do not produce any change. Okay, this alpha and beta, they only produce some change. Delta takes some time in production of these changes, right? After this we have biochemical test. So biochemical tests are performed to differentiate between the aureus and intermedius. So if we have to differentiate between the aureus and intermedius, we will go for the biochemical test. So in the Staphylococcus species itself, so what we will do, we will be requiring some purple agar, bromocresol, and 1% maltose. This purple agar is the medium, and this bromocresol purple is the pH indicator, and this 1% maltose is the substrate. After this, what we will do, we will grow the bacteria. If bacteria turns this solution into the yellow color, or it will form yellow color colony, then we will call it that this bacteria is Staphylococcus aureus, because they are good maltose fermenter. Okay, if there is no change in the color, then we will call it this bacteria is Staphylococcus intermedius. Why this yellow color production is there? Because these are good maltose fermenter, and they will produce the acid, and there will be the change in the color because we have pH indicator in this. So by this you can differentiate between the Staphylococcus aureus and Staphylococcus intermedius. Next test we have PCR, and this PCR is generally used in the reference laboratory or in the research. So it is not commonly used, right, to differentiate between the species. After this we have Coagulase test, and this Coagulase test is used to differentiate between Coagulase positive or Coagulase negative bacteria. Coagulase positive having the coagulase enzyme, whereas coagulase negative do not have this coagulase enzyme. Simple. And this can be done in two ways: slide test, and the next is the tube test. In slide test we detect the presence of bound coagulase, okay, but whereas in tube test we detect the presence of free coagulase. There are two type of coagulase enzyme. The first is the bound coagulase, next is the free coagulase. So we will detect the free coagulase by tube test. This is also important, okay, and we will detect the bound coagulase by slide test, okay. And in the slide test there is clumping factor on the bacterial surface, and this free coagulase we can call it staphylocoagulase. This clumping of bacteria appear in 1 to 2 minutes, and they are secreted by bacteria into the plasma. What this coagulase basically do, it convert the fibrinogen into the fibrin. So this is the major function of coagulase, okay. After this we have coagulase positive staphylococcus and their clinical importance, like Staphylococcus aureus. Staphylococcus aureus cause mastitis in cattle. It causes mastitis and tick pymia in lamb. Basically they are causing different disease, but we are talking about the main disease only, okay. They are cause a number of disease, but we are only talking about the main disease. And in the main disease you should learn 2 or 3 very importantly. Like in cattle they cause mastitis, in sheep they cause tick pymia, very important, this tick pymia, in lambs especially, not in adult one, right. In pigs Botryomycosis, and in dogs suppurative condition, scarus cord in horses. So these terms are very important, okay. And in goat they cause mastitis, dermatitis. In case of poultry they cause arthritis, septicemia in turkey, okay. So this is the general things. And after this we have next species, Staphylococcus intermedius. Staphylococcus intermedius cause so basically this pathogen is associated with dogs and cats, and this cause pyoderma, endometritis, cystitis, otitis externa, and suppurative conditions in case of dogs. And in case of cats they basically cause the pyogenic conditions. The Staphylococcus, this is very important. Staphylococcus aureus, it causes exudative epidermatitis, which is also known as greasy pig disease. This greasy pig disease is very important. You should remember this term, greasy pig disease. So greasy pig disease is caused by Staphylococcus aureus, and greasy pig disease is also known as exudative epidermatitis, and there will be arthritis also in this case. Next is Staphylococcus scleriferi subspecies coagulans, which cause otitis externa in case of dogs. Next we have pathogenesis. In pathogenesis, there are some structural feature like capsular polysaccharide, tricolic acid, protein A, interfere. Basically, they all these things will interfere with the opsonization and phagocytosis. And this opsonization and phagocytosis, this is a general process of a body to eliminate the pathogen. And if these things will interfere with these general process, then absolutely the pathogen will not be removed from the body. Okay, this cell wall protein bind to the fibronectin and fibrinogen, which will facilitate the bacteria to attach to the tissue. So by this, the bacteria will lodge in the body, and tissue damage will occur by the toxic factor which are released by the bacteria. So this is basically, in short, pathogenesis of Staphylococcus genus. Next, we will talk about the pathogenicity. So what is pathogenicity? Pathogenicity is nothing but it is the ability of an organism to cause disease. So how much a pathogen is pathogenic, you can say. So its pathogenicity is ability of that pathogen to cause disease. Okay, so Staphylococcus cause a number of diseases in different species, as we have read earlier also, right? Next, we have several virulence factors of Staphylococcus aureus, which are responsible for the virulence of these bacteria. The first is the coagulase. We have some pathogenic effect of coagulase, like it will convert the fibrinogen into the fibrin. So what is the harmful effect of fibrin formation in this? Because this fibrin will shield the bacteria from the phagocytic cell, and you know, if this fibrin shield the bacteria, these bacteria will not be phagocytized. This is obvious that these bacteria will not be phagocytized by the phagocytic cell, and if they will be not phagocytized by the phagocytic cell, they will remain in the body for longer period of time and produce toxins and harm our body. So this is the function of coagulase. Okay, after this we have lipase, esterase, elastase, and staphylokinase DNase. There are different enzymes which are produced by these bacteria, hyaluronidase and phospholipase. So these enzymes contribute to the virulence of the bacteria. And after this we have protein A. Protein A, basically it binds to the Fc component of IgG and it inhibits the opsonization. So you can see that these bacteria are majorly affecting the phagocytic process, which is the first one, and the opsonization process. Okay, next is leukocidin. It basically causes the cytolytic destruction of the phagocytes. So they are also attacking the phagocytic property of these cells of some animal species. Next we have alpha toxin or alpha hemolysin, and they are the major toxin in the gangrenous mastitis. In gangrenous mastitis, basically what they do, they cause the spasm of the smooth muscle, and after that there will be less blood supply to that organ or the mammary gland, and there will be necrosis. Due to this, the mammary gland may slow off in chronic cases, and it may be lethal to the animal. Next we have beta toxin or beta hemolysin. In beta toxin, basically this beta toxin or beta hemolysin, they act as sphingomyelinase and they damage to the cell membrane. Next, seventh we have enterotoxin, and this is also important. You will study this portion in detail in the public health and epidemiology portion. Okay, so they produce several enterotoxin which are heat stable toxin, and they cause food poisoning in humans. Okay, so they produces the heat stable toxin. This is important point. After this we have toxic shock syndrome toxins, and they induce excessive lymphokines which produces the tissue damage. Next we have differentiation of gram-positive cocci. How you can differentiate the gram-positive cocci, because there are different you can say pathogen apart from Staphylococcus. You have to differentiate the Staphylococcus from the Micrococcus, Streptococcus, and Enterococcus. Okay, so you can differentiate these gram-positive cocci as follow. The appearance in the stained smear. The Staphylococcus appear as irregular cluster. I will show you some images that you can relate. Okay, so they appear as irregular cluster, whereas Micrococcus appear as packets of 4, and in Streptococcus they form chain. If I talk about the coagulase production, then yes, Staphylococcus produce coagulase enzyme, whereas Micrococcus and Streptococcus do not produce coagulase enzyme. This is very important differentiating feature. After this, we have catalase production. So yes, Staphylococcus and Micrococcus produce the catalase, whereas this Streptococcus or Enterococcus do not produce. After this, we have oxidase production, O and F test. O and F, O refer to oxidative and F refer to fermentative. So basically these are fermentative in nature, they are oxidative in nature, these are also fermentative in nature. Bactricin disc, they are resistant, they are susceptible, and they are also resistant. So by these tests, several tests, you can differentiate between the Staphylococcus, Micrococcus and Streptococcus. Okay. After this we have several diseases. So we will discuss in brief about these diseases. The first is the bovine Staphylococcus mastitis. This bovine Staphylococcus mastitis is also important, and it is usually caused by Staphylococcus aureus. Remember the etiological agent. Okay, this is very important. Okay, so bovine Staphylococcus mastitis is caused by Staphylococcus aureus, and it may be subclinical, acute, or chronic. In majority of the cases, the mastitis is subclinical, and there will be peracute and gangrenous changes, and it may be life-threatening. In gangrenous mastitis, affected quarter will be cold, blue-black. It is cold or blue-black due to the less or cut blood supply. Okay, and it may slow off. There will be the alpha toxin, and this alpha toxin produces the tissue necrosis, and due to this tissue necrosis there will be and this alpha toxin also produces the contraction and basically necrosis of the smooth muscles, and this will impede the blood flow. And due to this impending of the blood flow, there will be cold and blue-black color, and there will be slowing of the organ. This slowing is the detachment of that organ from that area, from its natural position. Next we have tick pymia. In tick pymia, the infection of lamb is caused by Staphylococcus aureus, and it is confined to the hill grazing region. So it is not common in other region, it is confined to hill grazing region. Why it is common to only hill grazing region? Because it is caused by a carrier, which is a tick, and the name of the tick is Aedes ricinus. This is very important. Why this is only confined to hill grazing region? Because this tick is only found there. Okay, so it is in Britain and Ireland, and the minor injury or tick bite, so due to the minor injury or tick bite, this may cause the infection. Okay, this tick pymia may also be caused by Ehrlichia phagocytophila, because this Ehrlichia phagocytophila cause the immunosuppression in lambs, and if the lamb is immunosuppressed, so this will help the Staphylococcus to establish the infection in that lamb. Okay, and it is characterized by rapid death and septicemia. The treatment is prophylactic treatment with tetracycline, so the prophylactic measures should be taken. This is the treatment of disease. Next we have exudative epidermatitis, which we know that it is caused by Staphylococcus aureus, and it is highly contagious, and up to 3 months of piglets are affected with this exudative epidermatitis. It is also called as greasy pig disease, and this point is very important, and it is characterized by excessive sebaceous secretion, exfoliation and exudation, and it basically enters through the bite wounds, and the treatment is systemic antibiotic therapy. Last but not the least, we have Botryomycosis. It is chronic suppurative granulomatous condition caused by Staphylococcus aureus, and it is it happens when there is castration of the horses, and there will be infection of the stump of spermatic cord, which is known as serous cord, and the lesion in this case is mass of tissue become foci of pus. There will be mass of tissue, and there will be foci of pus. So these are the conditions which we will see due to this Staphylococcus. Now we'll look at some important images of Staphylococcus. The first image, as I have already discussed, that the Staphylococcus is present in the form of bunches of grapes. So you can see that this Staphylococcus form bunches of grapes. This mimics the bunches of grapes, right? And you can also see here that the Staphylococcus look like a bunch of grape. Okay, and this picture also depicts the same thing. Next we have Staphylococcus aureus. As I have discussed, the Staphylococcus aureus produces double hemolysis, right? And this double hemolysis is produced by two type of hemolysis or toxin. The first is alpha and the second is beta. as we have discussed that alpha toxin produce narrow zone of hemolysis, and this narrow zone is of complete hemolysis, right? It means that alpha toxin produce narrow zone. This zone is narrow, but there is no diffuse part. This is complete hemolysis, okay? Whereas in this opposite to this, the beta toxin or beta hemolysin produce wider zone, and there is partial hemolysis. Means you can differentiate between these two areas. This area is more diffused, but this area is more you can say that complete hemolysis is there in alpha hemolysin, okay? So this is known as double hemolysis, and this double hemolysis is produced by Staphylococcus aureus. This is very important, okay? So this double hemolysis is produced by Staphylococcus aureus. Two type of hemolysins are involved, alpha and beta. Alpha produce narrow zone, complete hemolysis, whereas beta produce wider zone and partial hemolysis, okay? After this we have generally we have three type of hemolysin, alpha, beta and gamma, but this gamma is not very important, and we generally discuss about the beta and alpha. Alpha produce narrow and complete. You can see the narrow zone of hemolysis, but it is complete, whereas the beta hemolysin produce wider zone, but it is partial, okay? So after this we have Staphylococcus aureus colonies, and if you look at the Staphylococcus aureus colony, they look golden yellow color, you can see in the plate. And and after this we have image of Micrococcus. So these are two pigmented colonies of Micrococcus species, and Micrococcus species can be white in color or they can be yellow in color, okay? So you have to differentiate Micrococcus species and Staphylococcus from the Micrococcus. You can see that this is the colonies of Staphylococcus intermedius, but it is overgrown by the Proteus species, okay? There is swarming growth, you can see, but this culture 112 figure, this culture is almost pure culture, and the both cultures are they are cultured on the sheep blood agar, okay? And as we know that Staphylococcus intermedius produce both type of toxins, alpha and beta. So you can see the hemolysis in this slide. After this we have Staphylococcus hiacus. As we know that Staphylococcus hiacus is a non-hemolytic, right? This is non-hemolytic on sheep blood agar. So you cannot, you see that there is no hemolysis on this plate. You can easily differentiate this plate and this plate. There is hemolysis, this portion is hemolysis, okay? This portion is hemolysis, but there is no hemolysis around the colony on Staphylococcus because Staphylococcus hiacus is non-hemolytic in nature. This is very important, that Staphylococcus hiacus is non-hemolytic, and you can see that there is no hemolysis around the colonies, but here is hemolysis. After this we have Staphylococcus aureus gram stain smear. You can see that these are Staphylococcus aureus in gram staining. Staphylococcus intermedius looks like this in gram staining. And after this we have DNase agar, Staphylococcus epidermis and Staphylococcus intermedius. This one is Staphylococcus intermedius and this one is Staphylococcus epidermis. Staphylococcus epidermis produces negative test in this case, and means they do not have the DNase activity, they do not produce the DNase enzyme, right? But Staphylococcus intermedius do produce, and they have a positive effect on this. Next we have purple agar base, in which 1% maltose is present, and as we already discussed that Staphylococcus, this is Staphylococcus intermedius, this is Staphylococcus aureus, and this is Staphylococcus hiacus. So there is change in color, and you can differentiate by this. There are several biochemical tests, okay, by which you can differentiate between the species. After this we have a tube coagulase test, and which we already discussed that this is a positive tube coagulase test because there is coagulation in this, and this is negative because there is no coagulation. And now we will discuss about some images of disease, so you can easily remember those disease. The first is the bovine Staphylococcus Mystitis. In bovine Staphylococcus Mystitis, we have generally gangrenous Mystitis, and the affected area is blue-black in color. So this is very important. The affected area is blue-black in color, and this is gangrenous Mystitis, okay? And this may be life-threatening to the animal. Not always, but sometime. Next we have tick pyemia in lambs, and in tick pyemia, there are no very characteristic symptoms in case of tick pyemia, but yes, there is lameness in case of lambs, and there is sudden death, okay? And the tick involved in case of tick pyemia in lamb is this Aedes ricinus. And after this a greasy pig disease. This greasy pig disease, you can see in greasy pig disease there is excessive sebaceous secretion. You can see the pig face is excessive sebaceous secretion is present on his face, and this is known as greasy pig disease. And yes, so these are some images of diseases which are caused by Staphylococcus bacteria. So this much in this video. Thanks for watching this video. If you like, then please press the thumbs up and subscribe to Vet Study.